Good afternoon, everyone. I'm Xinting, Field Application Scientist from GenScript Europe. GenScript is a world-leading biotech company providing life science services and products. With gene synthesis, peptide, protein, antibody, and preclinical drug development service capabilities, we are internationally recognized as a leading biotech company specializing in fundamental life science research and early-phase drug discovery services. So next, it's my pleasure to introduce our speaker, Dr. Leo Fan. Dr. Fan is the Senior Manager of Bioinformatics team at GenScript. He is also one of the inventors of GenSmart Codon Optimization algorithm. Today, he wants to introduce how to empower protein and antibody expression by GenSmart Codon Optimization. This webinar will take around 45 minutes. You can type in your questions in the chat box, and Dr. Fan will answer them at the end of the webinar. If you didn't get your answers during the Q&A section, we will take them down and email back to you. So without further ado, let's welcome Dr. Fan to start the presentation. Dr. Fan, over to you right now.
Thanks for Xinting's introduction. Good afternoon, I'm Leo Fan, the Biological Big Data Intelligence Analysis Office Manager. Today, I'm glad to share how to utilize the codon optimization to improve the expression of protein and antibody. So this presentation includes 3 parts. Firstly, I would like to address the difficulties we are facing during the protein expression. Then I will show our comprehensive validated solution. Finally, I would like to show some case. So when we discuss with our clients, they always ask, why my protein yield is so low, even if the sequence were optimized by some academic tools? And then would like to ask how to balance the codon usage when switched to a new host species, or learn from the literature that it's better to remove some motifs that might have negative impact on my protein expression. But how can I do it personally? And then they would like to ask, the expression yield is good, but the function protein are not sufficient for the subsequent functional assay. So is it possible for me to change this situation? So before providing the solutions, I would like to recall what we learned from the textbook before. The first step of protein expression is the transcription, which involves the phase regulation element, the site having effect on the chromosome stabilities, such as the G site and the polymerase sleeping site in E. coli. The mRNA processions and the stabilities is also the key step. So which influence the lifetime of mRNA? The factors contains the cryptic splice site, the mRNA secondary structures, stabilities of free energies of mRNA, so and the GC percentage and the CG associated immunogenetic. So then the translations efficiencies will be influenced directly by the codon usage bias, the ribosome binding site, and the premature poly A size. So finally, the functional proteins required collect and efficient protein foldings and the modification, which could be influenced by the codon context, the codon anticodons interactions, and the translation port site. And the most question from our customers is that since there are so many factors here based on your experience and research, which factor is the most important and can you rank them? So is there any answer for these questions? Actually, the scientists have no idea why. Obviously, the protein expression is really complicated, even focus on the same factors such as the CI. Different groups have no consensus conclusions in various expression systems. and it might have different influence on different proteins. When one codon change, the value of a group of factors change together simultaneously. That all of the factors are not complicated independent, and it's hard for us to evaluate their effects from single site. However, the identical observation is the existing of codon bias within or between the genomes. Here you will find for the different species, they have their own preferred codon usage patterns shown in the heat map. The red is a preferred codon, and the black is the rare codon. So some reviewers also help us to summarize the reported key factors which have influence on the expression, such as the CAI, the rare codons, and the mRNA folding. But even they say the high GC content or the CpG content are preferred by highly expressed proteins in mammalian systems. This is only the trend, but it's not the absolutely right for every case. So another review paper not only listed the name of some factors, but also their correlations with both protein abundance and mRNA abundance. And you will find that some factors only have a significant effect on the mRNA levels, and some factors only have a relationship with protein's abundance. So it demonstrates that various parameters have different story and play different roles, and it's not easy to conclude is it significant or not. So when should we perform the codon optimization? This is a general workflow of the protein expression, as you might see several times before. The codon optimization is manipulate of the DNA. So we will optimize the sequence first, and then the optimized sequence will be synthesized and then insert into the expression plasmid. And then we will do the purifications, the QC, and to get different proteins for the further applications. So far, the codon optimization target has been widely used in many applications. For example, the recombinant antibodies has been utilized to accelerate the antibodies drugs development. The recombinant proteins has been used in vaccine development as the antigen or other key raw materials. So in addition, codon optimization has been play important role in the production of two enzymes. A good example you might be familiar with is that the Cas9 proteins and the Cas9 proteins could be used for the gene editing. So in other words, the codon optimization together with some other technologies has facilitated the biological research. So, but unfortunately, we are still facing some challenges. The raw expression of some target, the solubility and the activity issues, even the immunological response, either or both resulting the function issues. So we will talk about the solutions, what we can provide. When we talk about the proteins expressions with the clients or the colleagues, we will discuss the constructions of expression vectors, and those vectors consist of the backbone of expression vectors and target insert. The insert could be manipulated at the DNA levels by applying the codon optimizations. For different purposes, we can select the different host cells. And in addition, the protein tags provide important roles in the solubility or the purifications and others like the reporters as I listed here. So my colleagues might introduce the host and the tag before in the future. But today I will focus on the manipulation of DNA levels. So I will share how to improve the protein and antibodies expressions. And I would like to elaborate the algorithm behind the tool which we launched for free usage. So before moving to the explain of the algorithms, I would like to take this opportunity to review the evolution of our algorithms through the iterative refinement. So in 2008, we launched our first generation algorithm based on the particle swarm algorithm. In 2015, we upgraded the algorithms for the E. coli host, followed by the performance improvement at the mammalian host systems. A patent based on the immune algorithm has been applied. A big milestone was achieved in 2017, and we formally launched the GenSmart Codon Optimization tools in 2018 based on the newly developed algorithms. And it's based on the multiple objective genetic algorithms. And this tool has covered over 50 expression hosts. These 2 years, we keep the algorithm moving forward, and we had completed a lot of customized requests, such as to make the GC percentage maximum, to make the number of CPG dinucleotide minimum. And we also help the customers to optimize the sequence and make this sequence to enable optimize sequence to define the sgRNA recognitions and so on. So what the algorithms for the GenSmart Codon Optimization tools? The principle is to join on the advantage and avoid disadvantage. Firstly, we collect the highly expressed genes and done the data mining, and then we will calculate their characters such as the codon frequency, the codon pair frequency. Thirdly, we will utilize the genetic algorithms to optimize those 3 objective functions, including the harmony index, the codon context index, and the outlier index. And please refer to our patent about the definitions of 3 parameters. Finally, the harmful motif should be removed, and those harmful motifs were collected from the publications, or could be the customized input by our customers. The backend genetic algorithm is inspired by the process of natural selections, and this algorithm belongs to the evolution algorithms. In this algorithm, the first generations of randomly generalized sequence will go through the population evolutions, like mating, mutations, and selections, and all of their fitness will increase. And the fitness means the value of 3 objective functions, as we mentioned here. So with the help of GenSmart Codon Optimization tools, we aim to help you to get more activities proteins for your further study. Right now, the most common used species has been covered and is free, and the interface is user-friendly. As I mentioned before, we done some collaborations with the customers and complete some customized request. And all factors mentioned above associated with the protein expressions will be taken into the consideration. Yeah, as the third part, I will give some case studies which have been applied our algorithms to their studies. Firstly, I would like to share the data we present in our patent. We access the performance of GenSmart Codon Optimization tools in increasing the expression of GNK3 and the GFP proteins. And we will using the beta actins as the internal control, positive control. And we measured the proteins yield of 3 different clones in the control systems through the Western blot. And as you can see from this chart, both the GFP proteins and the GNK3 proteins show a significant increase in the protein expressions, respectively. So they have obviously highly expression than the wild type. So it has been reported that the codon optimizations could help to improve the expression of recombinant therapeutics, and the expression can increase over 1,000-fold after only one codon modification. So the data here we show are for the 20 recombinant antibodies in the control systems, and we observe the continual increase of the latest versions when compared to the previous versions of 2 years ago. And then I would like to show some tests in the publications. In 2017, Dr. Liang from the NIH using our algorithm for the optimization of vectored vaccine. The graph is cited from Dr. Liang's patent. You will find, compared with competitors' algorithm, the gene optimized by algorithm expressed highly than our competitors in both the vernal cells or in the LLC MK2 cell. So, and what's more important, our optimized sequence cause less immune reaction. In 2018, the David Liu, a famous researchers in the genome editing field, also using our codon optimization tools to optimize the nuclear locations codons of BE4 base editors. And from this graph, you will find the GenScript or the yellow column. The GenScript assures the best performance evaluated by the raised number of the NGS result. And we also found a lot of independent data to demonstrate that codon optimizations could help to improve the expressions at the fungi, E. coli plant beside the mammalian systems. Here we just select 2 citations as an example. Our algorithm helped the antibody extract productions and the COVID-19 proteins optimizations. And here, the 2 cases were used for the gene therapies and metabolic engineering. And the customers done some comparisons between different algorithms and tools from various vendors, and it demonstrates that the GenSmart Codon Optimizations work best through the Western blot or the titer test. But it's a pity that we can't share all cases that our GenSmart Codon Optimization has facilitated a lot of study here. But it's exciting that it has been globally recognized and widely used in the academic and the industrial field, including some famous big pharma. So this slide shows you how to use the online interface of GenSmart Codon Optimization tools. We mentioned it's so easy and it's user friendly. Firstly, you need to copy and paste your sequence here. And, then you will need to select your species from the drop down list, and then just one click to submit your task and waiting for the result. By the end of my talk today, I'm pleased to introduce more GenSmart modules here. In addition to codon optimizations, we are still waiting for launch some new modules. We developed the GenSmart designs for you to design your vectors, and we have online quarter and ordering systems called GenSmart Instant Quarter, so you can get your price within 1 minute. You also have, and we also have GenSmart variable domain sequence analyzers to analyze the sequencing date of antibody library, including both the first generation or the second generation sequencing date in batch mode. And other tools such as upgrade primer designs, the hybridization probe designers, and multi-pass primer designers are waiting for launch. All those back-end algorithms have been validated by our internal wet lab. So hope this advanced tools could make your research easier. So thank you for your listen, and I'm happy to take your questions.
Okay, thank you, Dr. Fine, for the great presentation. And everyone, if you have any questions about the presentation, please type in your questions in the chat box. And now let's move to the Q&A section right now. Dr. Fan, here are some questions, and then I will ask them one by one. So let's move to the first question. Compared with other codon optimization algorithms, what are the advantages of GenSmart codon optimization algorithms?
It has been demonstrated by other researchers in different fields that GenSmart codon optimization algorithm could boost higher protein expression and decrease the immunoreactions. And I think this is why so many our customers cite our tools in their papers and even books. And that's why it's the big pharma companies ask for our offline customized codon optimizations as well. Yeah.
Okay, thanks, Dr. Fine. So the next question, how do you come up with applying the genetic algorithm to codon optimization algorithm?
As you know, the immune algorithm is inspired by the human's immune systems, and this algorithm could increase the diversity of populations, or we say the diversity of antibodies, and it could avoid the populations derogations, and so it's benefit to improve the capacity, and so the algorithm could output the global optimized output.
Okay, thank you, Dr. Fan. So the next question, would you like to elaborate a bit on why did you identify these 3 factors for GenSmart Codon Optimization algorithms?
Oh, I think it's fortunate because we do a lot of experiment, and we also try other parameters, but we can't completely recover or redemonstrate the results shown in some papers. So that's why we present the 3 3 novel index, and we using those 3 index to generate some suitable output, and this optimized sequence show higher expressions level compared to the wild type, and even in the comparison with our with other vendors' solution.
Okay, thanks, Dr. Fan. So the next question, I want to optimize for other species outside of these 50 species. Can the GenSmart Codon Optimization algorithm support it? Do I need to provide any additional information?
Sure. We have already helped a group of our customers to do the offline optimizations, and the customers could provide the species name, but we hope those species have well annotated genome informations. Yeah.
Okay, thanks, Dr. Fine. So the next question, what do you think the next step in the development of codon optimization algorithms?
Suppose we can generate a big mutation libraries, and we do the experiment to get those expression data, and then we can using the artificial intelligence method to do the data mining. But whether we could achieve better improvement is uncertain, but because it will also cost too much the labors and the money. Yeah.
Okay, thank you, Dr. Fan, for your detailed answers. I think we have already answered all the questions from the audience. So we just end the Q&A section here. If everyone, you have any more questions about this webinar or the codon optimization tools, please contact us by email. We would like to provide more technical support. And then we will update the recorded video on GenScript website for review and share the link with you via email tomorrow. I hope you find this section useful and beneficial for your work, and we will continue to launch more webinars on various topics. So please follow us in LinkedIn to stay tuned for the latest webinar schedule. And thank you to everyone for joining again, and thanks Dr. Fan for the great presentation. I will.
My pleasure, yeah.
Thank you, Dr. Fan. And I will conclude this webinar right now, and have a nice day, everyone. Bye-bye, everyone.
Bye.
Bye-bye. Fine. Bye.